This afternoon, we come to a very special part of the HLF, and it's one of my favorite parts we have each year. It's the Lindau lecture. There's a close connection between the Lindau Nobel Laureate meetings and the Heidelberg Laureate Forum, and that the Heidelberg Laureate Forum, in a sense, was designed after Lindau, and we really learned from Lindau. So Ruth Wetzlar, our managing director, the year before the first Heidelberg Laureate Forum, she went as an intern to the Lindau meetings to learn how things are done. Then, of course, it's not the same. Some things are a little bit different, but these close ties are always there, and we express them by having every year HLF laureate give a Heidelberg lecture in Lindau, and this year was Jack Dongara and also Yael Kalai, and having a Nobel laureate come to us and give the Lindau lecture. And I think it's so great to have the Nobel laureates come here and also to tell us about something which is usually not math and computer science, but I mean usually very, very interesting, sometimes perhaps even more interesting than, because we don't see it so often, right? So, but, so this is now what we are going to hear, and it's my real pleasure to that we have David MacMillan, who won the Nobel Prize or received the Nobel Prize in Chemistry in 2021, together with Benjamin List, for the development of asymmetric organocatalysis. And he will tell us about the pathway to discovery in catalysis. David, it's a pleasure.
Okay, thank you for that kind introduction. Thank you all for coming to this lecture. I realize this is somewhat of a departure from what the rest of the conference has been. I will say for myself, from my own point of view, this has been fascinating for me, both from mathematics, computer science, and maybe even sociological reasons, just to sort of see how scientists in different areas interact with each other, which has been really, really fun. The talk I'm about to give, I actually gave it at the Lindau lecture earlier this year, and Jack Dongara came up to me and said, this would be great for the HLF. So if you don't like this lecture, it's not my fault, okay? Blame it on Jack, all right? Second thing is, I'm going to tell you about what we worked on to win the Nobel Prize for about the first third of my lecture. And then the second and third part is going to be things that we've done subsequently, because it was really work that I did in the first 10 years of my career. And I'll explain what it is, but then I'll hopefully demonstrate what it led to and where we're going with it. Okay, so basically, if it shows up, asymmetric organocatalysis. This 2021, as mentioned, I was very lucky, very fortunate to find out I'd won the Nobel Prize. And since winning the Nobel Prize, people ask you lots and lots of questions. And one of the main questions that people always ask me is organocatalysis. What on earth does that actually mean? What do these words mean? So I'm going to break it down really quickly, and I apologize if some of you know some of these things, but I always think it's good to start off fundamentally. And so what is catalysis to begin with? And one of the ways I explain this to everyone I ever meet is, if you look around you right now, absolutely everything that you can see is made by a chemical reaction. And this is a photograph or a picture of my office in Princeton, and absolutely everything in it is made by a chemical reaction. I realized looking at these slides earlier, this computer screen is the only reference to computers in this whole talk. So I apologize for that. Anyway. Yeah, so it's so funny. My wife thinks this is disgustingly messy, you know. All right. But so I didn't know there was going to be heckling during this talk, actually, but I kind of like it, actually. It's pretty good. All right, so, but basically, if you look at this, everything's made by a chemical reaction. Now, I can highlight that just by going through. Here are all the various chemical reactions that are used to make all these variety of different things. So if you hone in on one of these, which is my coffee cup, which is hiding back there, and this is the chemical reaction that makes caffeine, the stimulant in coffee. And most people know in chemistry that chemical reactions require energy. At room temperature, most of them are not spontaneous. And the way that we can demonstrate that is we can show it in an energy diagram. And I'm not going to get too technical. This is the way we show an energy diagram of the starting materials becoming caffeine. Now, when I talk to high school students, the way I explain this from a metaphor is, for example, if I had to walk home every single day and I had to walk over a pretty large hill to get home, that would be inconvenient and it would involve the use of a lot of energy. To overcome that, what you can do is you can build a tunnel through that hill. And that's exactly what catalysis does. It builds a tunnel through the hill. So it makes it much easier to get to your destination. And that's what it does in a chemical reaction. So if you take anything away from this talk, just memorize the fact that catalysis makes existing chemical reactions easier and faster. But the most important point is it allows chemical reactions that were previously impossible to become possible, which is really the critical factor. Okay, now if you're wondering, does catalysis really impact our world? There's many, many examples, and I'm just going to show you my favorite one. This is the global population of the Earth over the last 2,000 years, and it's pretty much flatline. Gets to the beginning of the 20th century, there's an inflection point, and it races up to 8 billion people on Earth. And as it happens, you couldn't have 8 billion people on our Earth if it wasn't for this one catalytic reaction, the conversion of nitrogen into ammonia. And if you're wondering why do we need ammonia to have 8 billion people on Earth? You need it to make food. You need it to grow crops. Without all of that ammonia, we couldn't feed 8 billion people. So one of my favorite little factoids about this is if you actually think about your body right now, and if you think about all the protein, the DNA, and the enzymes in your organs, and all those nitrogen atoms that are in all of those molecules, 50% of them come from a factory in Germany. If that doesn't freak you out, nothing will, all right? So I think that's just incredible, actually. All right, these are some other statistics. 90% of industrial scale chemical reactions use catalysis. 35% of world global GDP is based on catalysis right now. And as we think more about sustainability and climate change, that number has to go up. It certainly has to go up. So catalysis is important. Second thing, what does organo mean? Well, I'm going to take you back to 1998. I'll explain why in a few moments. But in 1998, what were the main areas or branches of catalysis? There's really just two. So one of them is biocatalysis. This is using the catalysts of life, which are the enzymes, for example, in all living creatures. It's kind of interesting, actually. I was reading about this last night, that Januvia, which is a type 2 diabetes medicine, is actually made using enzymes that come exclusively from the American bald eagle, believe it or not. They don't actually use bald eagles to make the medicine, but the enzyme itself was discovered from that, which is pretty remarkable. The second, but even larger area is metal catalysis. And metal catalysis is completely human invented and designed and developed in laboratories. And in 1998, these were the two major areas of catalysis. Okay, so metal catalysis. So if I show you a metal catalyst, even if you're not a chemist, you can look at that molecule and realize there's really two parts to it. There's a left-hand side and a right-hand side. And if you separate the two parts, you see on the right-hand side is the metal center. Now, it's often the case that metals are expensive, they're toxic, they can be very difficult to work with. You have to use glove boxes, which means you have to shove your arms in these boxes for 8 hours a day to work with them. They also have a problem with sustainability. For example, palladium, which we have in our iPhones or in our catalytic converters in our cars, we've been using palladium on Earth for about 90 years. But we only have 90 years left of palladium on Earth, and it will basically be dispersed, and we won't be able to use it anymore. Completely not sustainable. But the left-hand side of this catalyst is the organic framework which holds on to the metal. It's not usually the business end. It's not usually the part that does the chemistry. But the organic molecules are everywhere on Earth because they're the molecules of life. All living creatures, all living aspects of life have organic molecules. So they're typically inexpensive, safe, sustainable, because when we pass away, we go right back into the life cycle. We get completely recycled back out again. So based on this, I started to think, why are we using metals to do catalysis? Why don't we just use the organic part as the catalyst? Now, this eventually became known as organocatalysis, not at that moment, but that's the area that we're going to talk about. So why 1998? Well, 1998 was when I landed a job as an assistant professor at Berkeley. And this is my first class, my first lab in our first year. This is our first year at Berkeley. And I really love this photograph because it's Friday night, it's 10 past 10 on a Friday night, and we're in there working hard, trying to make an impact, trying to make a difference. So what were we going to work on? Well, I decided I wanted to work on this concept of using organic molecules that didn't have this name yet. And the reasons, if I haven't pointed to them so far, are really straightforward. These molecules are everywhere on Earth. They're really readily available. They're not sensitive to water and air, much like we're generally not sensitive to water and air. Because they're so abundant, they're very inexpensive. You don't have to use these glove box contraptions to work with them. You can do it out on the open. And they're completely sustainable. They're non-toxic, and they're recyclable. And these are all really good reasons, but it turns out this was not really the reason I wanted to do this. I was really fixated on the idea, wouldn't it be great if you could take these organic molecules and get them to do not just one chemical reaction, but maybe do 10 or 100 different types of chemical reactions, which would potentially be a field. The problem was that was a very grandiose idea, but I had absolutely no idea how to do it. Zero ideas on how to do this. But I was working in lab one day, and I was in lab, and this is one of my first graduate students in the first year. His name is Tristan Lambert. He's now a lot older, and he's now a very successful chemist. He's now the chair of the Cornell chemistry department. And he came up to me as a first-year graduate student, asked me a really simple chemistry question. He said, what is the mechanism of reductive amination? Being a young professor, I sort of ran to the board to tell him what we were doing, what this means, and I drew up this mechanism. And you don't have to know chemistry to know that there's two molecules on the left become one molecule on the right. And it's only when it's on the right-hand side, what's called an aminiamine, that has the right electronics to do the subsequent chemistry. And it was right there, right then, I had the quintessential eureka moment, because I suddenly realized, wait a minute, if you take this other type of molecule and do the same thing, this looks suspiciously similar to a whole field of catalysis that had been using metals for many, many decades, shown here, written by the letter M. Now, if I draw this in a much more conventional chemistry way, the equation on the top here is called Lumel-Loring metal catalysis, and it's literally been used for hundreds of reactions. The one on the bottom equation, I've simply replaced the M with an organic molecule, and there's basically no examples of using that to do catalysis. Now, this is my mathematic part of my talk. So if these are simultaneous equations, see, I got that in there. If these are simultaneous equations, I guess that's algebra. If these are simultaneous equations, that really means that you potentially could use organic molecules to do these hundreds of reactions that previously had been demonstrated with metals. Okay, so we had the idea. We then had to decide, well, which reaction are we going to test this on? So we decided to pick probably the most famous organic reaction in the world, which is known as the Diels-Alder reaction, and it's such a powerful reaction, it won Diels and Alder the Nobel Prize in 1950. And it is powerful because it allows you to take relatively simple molecules and make much more complicated ones in a predictable fashion, such as the one shown on the right-hand side. So we did that. We took exactly two molecules on the left. We're hoping to make a product on the right. This is the notebook of my first student who did this, Kateri. And over the arrow, there's not M for metal. There's now an organic molecule. And when we performed it, we go at the bottom of the page, it worked. And it says not racemic here, which meant it worked. So I always remember getting this result, and I thanked my student. I walked into my office, I closed the door, I locked the door, I closed the blinds, and then I jumped up and down for about 10 minutes. I was so excited. I remember calling my wife, and I said to my wife, I think we're going to get tenure, which when you're an assistant professor, that's all you care about, right? It's like, can we get tenure, can we get tenure? All right, so that was where we were very excited. All right, so then we had to publish the paper. And publishing the paper, I had to come up with a name for this. So we decided to call it organocatalysis, combining organic and catalysis. And if you're wondering what's in a name, is a name important? Naming things is really important. This is Johann Jakob Berzelius. He's a scientist from Sweden. In the 1800s, he invented a lot of the vocabulary we now use in modern chemistry. Things like catalysis, protein, polymer, isomer, organic, inorganic. He named all of these things. And naming things, the value of it sort of has traveled with us into the modern era. AI, machine learning, directed evolution, nanotechnology, organocatalysis, they provide a sort of name or a framework for people in the field to come together to talk about it. So it is important, I would argue. But what was even more, I think, important, we introduced a concept of a generic activation mode. That sounds very technical. What does that mean? It's just a really fancy way of saying this won't work for just one reaction. This is going to work for hundreds of reactions. I'm proclaiming this in my first paper, which you'll see in a minute was not a clever thing to do. But I'm saying this should work for lots of reactions. All right, so this was our first generation catalyst. And this is our first paper, the Diels-Alder reaction. And so we went off to show it's going to work for hundreds of reactions. And it went bang, bang, nothing. It stopped after 3 reactions. So at this moment, I'm starting to have panic attacks. I've just told the world, this is going to work for hundreds of reactions. We've got 3. So this is really embarrassing. But what was incredibly fortunate was this first generation catalyst, I had these 2 amazing young graduate students, Joel and Chris, and they came to me and said, look, if we change just where it says Me, that's a carbon. If we take that carbon out and put 3 other carbons around the outside, we predict this is going to be really great for lots of reactions. This is something you would call precision molecular engineering. And when I talk to non-chemists and use a metaphor, I say, well, think about the outcome we're trying to get to. It's a bit like a precision athlete trying to do a precision outcome. And this is Zlatan Ibrahimovic. I'm a huge soccer fan, football fan. So I was like, I put this in to demonstrate that. And one of his most favorite goals he ever scored, I love to show this, is this goal shown here. He does this overhead kick, and bang. That's an amazing, amazing goal, right? Now, I actually showed this in my Nobel lecture, and I'm from Scotland, and a lot of the newspapers in London got really, really angry. And the reason why they got really, really angry was they said the only reason MacMillan showed this is because the goal was scored against England, right? And I think that's really, really unfair, but let's watch it one more time. All right. Look at that thing. Look, oh my God, it's so good. All right. All right. There's nothing better than being on stage and people clapping a goal against England. You know, it's just, all right. So anyway, so the question is this going to work for us? Is this precision change going to work? And so that was our first catalyst. We went to the second catalyst and bang, it starts to really go. We're off to the races. Other people start to jump in, Jorgensen, Hayashi, lots of other people start introducing catalysts and it really takes off like gangbusters, which was amazing to see. All right, so what can you actually do with this? Why should the world care? So there's lots of different things. I'm only going to show you really 2 applications. One of them is kind of fun, and that is in fragrances, flavors and fragrances, perfume. Perfume is actually, it sounds not that substantial, but perfume is used on an enormous consumption basis on a daily basis around our world. And so Firmenich was actually able to make Lily of the Valley now in 1 chemical step instead of 6 chemical steps. And the energy savings on that alone was just absolutely enormous. But probably and easily the biggest application of it has been in medicine. And I'm not going to show you that many examples. I'll show you one. This is talsedupant, which is a migraine treatment. Migraines affect about 3% of the world's population. And Merck were actually able to produce this molecule using organic catalysis to actually build it out, which was something for me, which was wonderful to see. Now, there was another advantage that came from it that I have to admit I did not see coming, and that is something called democratizing catalysis. And it's basically the idea that this type of catalysis is incredibly cheap. It's incredibly affordable. And as a result of that, it's actually practiced on every continent around the world, not just the Western Hemisphere, it's actually used everywhere. So when people ask me, what is the next big idea coming from in organocatalysis, I always say, well, number one, I don't know. But the second thing is, I know that it's not going to be based on who has the most money, it's going to be based on who has the best idea, which for someone from my background and my values, I think is something which is really pretty cool. All right, so basically we have to think about this business, 8 billion people, the world continues to grow. What are we going to do for catalysis? And these are 4 areas. Organocatalysis is one of them, which is going to enable us to move forward, but we need more areas of catalysis. This is something I preach to young chemists everywhere. We need a lot more, especially for climate change, which is going to be absolutely critical. One of these is photocatalysis, and I'm going to tell you now about stuff that was After the Nobel part, this is now the work that we've been doing subsequently. And we've been really interested in sustainability and how do we develop new and sustainable types of catalysis, but now using photons. All right, now this is something that, to be perfectly honest with you, is an idea that I've completely stolen from inorganic chemists. And these are inorganic chemists who are worrying about powering the planet with solar energy using certain types of materials. And we became interested in, could we export some of those ideas into organic chemistry? That was a number of years ago. This has now become known as photoredox catalysis, and it's basically we're using visible light. This is blue light in these cases, which is enabling completely new chemistry to take place, which I'll explain in the next few minutes. All right, now, to try and convince you that this is taken off in chemistry, one of the ways you can do this is use a bar graph, and this is the sort of publication numbers in photoredox. And this is beginning at 2008 when we first started doing this, and now it's up to about 1,800 papers a year. So it really has exploded within the field of organic chemistry. And I'm going to try and explain to you or contextualize to you why this has really taken off. So the first part to understand is that most organic molecules do not absorb in the visible region. They're colorless. But these molecules that the inorganic chemists invented actually absorb in the visible region very selectively and have 60 kcals per mole of energy per molecule when they are excited. Now, this turns out to be really, really interesting for organic chemistry. So this is a flask. It's got showing some organic molecules in here. If we sort of zoom in on it, and there's the photocatalyst shown here. Now, this photocatalyst is surrounded by all these other organic molecules, but it will selectively absorb a blue photon. And when it absorbs a blue photon, it goes into the excited state. It basically moves an electron from the metal out onto the ligand framework. And when it does that, it basically is the equivalent of being at 32,000 degrees Celsius, while every other molecule in the vessel is still at room temperature. So they're now on completely different energetic planes. So what it's got to do is it wants to get rid of that energy. To get rid of that energy, what it's going to do is it's going to either give electrons to other molecules or take electrons from other molecules. That sounds really chaotic, but it's not. Because you can selectively, for example, give an electron to that molecule, and when you do it, it forms a radical. Now, for those of you who haven't taken organic chemistry, radical, like the name suggests, means it's very, very reactive. It will react with things very rapidly. It's an extraordinarily activated molecule. But we've made it from a molecule that had a really low energy to begin with. Now, that's given electrons. You can also selectively, if you want, take electrons, in this case, with loss of CO2 to generate a radical here. But the big take-home is you can use just simple blue light to basically switch on all these molecules into radicals that previously people thought were completely inert. And although they're highly abundant on Earth in lots of diverse structural format, people thought you really couldn't use them broadly. But now you can because you're using this little blue light approach to doing it. All right, so this was the first paper we published, was back in 2008. We published it the same day as Taeshik Yoon, who was a graduate student in my group. He's a fantastic scientist. Our paper, there's now about 100 papers in asymmetric calculation. Taeshik's on cycle addition. He's totally destroyed us. There are about 600 papers there. But it doesn't matter because there's now papers in lots and lots of different areas of chemistry that uses this principle broadly. Okay, so why would people be interested in it? Well, the number one industry that's interested in it is the pharmaceutical industry. And I'll try and explain why. So these were the types, or are many of the types of building blocks that pharmaceutical companies use to make drugs. And they use these because they can put them together in a modular fashion, much like the way you would do with Lego blocks. So if I was to think about using Lego blocks, these are the types of Lego blocks. But the one thing I'll point out here is they're all very similar to each other, these building blocks. So as such, they can put together drugs, and this is a drug that came about from these types of building blocks. But the problem is these types of drugs tend to be less selective, and they often have more problems, especially in the clinic, when they get into human beings. And the reason, if you really want to get into it, is because they're flat. They're not three-dimensional. They're really two-dimensional molecules. Now, photoredox, and was the other analogy here, so you can build, for example, a house, but you don't get a lot of complexity when you build these types of molecules. All right, now, photoredox allows you to take these very different types of molecules. These are three-dimensional molecules you can find everywhere, but now you can start in a modular way, plugging these things together, again, like Lego blocks, but now with very different shapes, very different three-dimensional shapes. And because of that, that allows you now to build, again, a house, but a very different type of house, right? A much more specific styled system. And that, if you bring it over to the drug context, allows you to make molecules that have a much, much higher chance of making it through the clinic, or basically being successful in human beings, because of the nature of the building blocks that you're actually using. As a result of this, every pharmaceutical company now uses it on a daily basis, I think, which is pretty neat. These are really all automated now. There's lots of chemists who use them, but there's a lot of what's called high-throughput synthesis, performing these with thousands and thousands of chemical reactions. Just to give you a tiny example of some of the things we've been able to make with this, this is from Lilly. This was a cancer drug. It's a JAK2 inhibitor. From Novartis, they were actually able to make DNA-encoded libraries. Gilead this is an anti-HIV drug that they made using photoredox. And Merck, this is pretty cool actually. Merck are making these kind of derivatives of insulin that have better properties than insulin by making chemical changes to them, and again, using photoredox to do this. And this is just to show, those were all on small scale, but once you start to go to a larger scale, you have to be able to scale up how you get all the photons into these chemical reactions. This is Merck process in Rahway, New Jersey, and some of the systems that they can stack back to back to back to do literally getting this up to ton scale synthesis as they actually make these molecules, again, using blue light. All right, so that's chemistry. I'm going to finish off in the last 13 minutes and talk about, can you gain insights into biology using photons and catalysis? And I'll explain why we get into biology in a second. But this is something that's become now known as micromapping. And this is the same photoreactors with blue light, but now you're shining them into basically Petri dishes with cells in them. Okay, so basically, I think most people understand this fundamental biological principle that in biology, we have proteins, we have DNA, we have all these biologic molecules. And it turns out that these proteins, for example, this biologic, this protein of interest, that in any given time frame, they're constantly interacting with lots of other biological molecules, such as other enzymes, maybe metabolites, small molecules, DNA, RNA, etc. Now, as a chemist, what was fascinating to me in talking to some of my biology friends was the fact that the vast majority of these interactions are just simply not known. I didn't know this. I thought most of biology kind of was known. It turns out that's not the case. We basically are scraping the tip of the iceberg, so to speak. So basically, people don't understand what all these different things are interacting with in a normal course of a healthy cell, for example. So one of the questions that comes up is, how do you identify all these different interactions? How do you do that? Now, there is a strategy, and we did not invent this. This was a beautiful strategy invented by an amazing scientist called Alice Ting. And the idea is, if you take a protein of interest, for example, but now you stitch a catalyst onto your protein of interest and let it go about doing its business, wherever it goes now, it will catalyze a chemical reaction and put a labeling agent onto all the things that it interacts with, and effectively leaving its footprints wherever it goes. And now you can use a simple pull-down method with proteomics to understand everything it interacted with. So all of that sounds fantastic. Now, the way that Alice did it was, this was a protein of interest, that dot shown there, and the catalyst that she used is horseradish peroxidase, which is stitched on to her protein of interest. And what she would do is she would use these molecules at the top that are basically going to be the labeling reagents. And she would expose them, and again, they would form radicals. This thing in the top right is a radical. And I've told you radicals are really reactive, so they're going to stitch themselves onto things. Now, there's a problem. The problem is these radicals actually have a reasonably long half-life, meaning they stick around for quite a while. Why is that a problem? Well, that's a problem. It means because they diffuse. Not only will they stitch onto the thing it's interacting with, they'll diffuse away and start stitching themselves onto lots of other things inside or on top of a cell. So basically the granularity of this is not very good. You get tons and tons of false positives, but it's good for things which are far away, for example, for organelles. But if you really want to get precise, you have to get down to about this 10 nanometer range, especially if you care about disease state chemistry or disease state biology. Okay, so we thought about this, and we thought, well, how do we map these? How can we come up with a way that's just so much more precise that you're only stitching these agents onto things which are right next door? And we realized this is a chemistry problem. The problem is the radicals they're using basically diffuse too far. They live too long, 100 microseconds. But as chemists, we knew that these carbines, these things shown here, have a 1 nanosecond half-life, which means that most they should travel up to 10 nanometers before they die, which is exactly what we're looking for. So based on this, we asked, well, how do we localize the formation of these really reactive species at any given spot in a biological system? And so that became the question, how do we actually activate these things? And the answer was pretty straightforward. We simply would take, for example, an antibody. We know that antibodies, this sort of bent thing here is an antibody, and the IR with a circle around it, that's a photocatalyst. And the antibody is very selective for whatever protein you care about. But the goal now is when you shine blue light on this, there's only one thing that's going to get excited, and that's the photocatalyst, because it's so selective at picking up the photon. And it can do a thing called Dexter energy transfer, and that can only take place up to 2 to 3 nanometers away. So that enforces this really tight radius around your protein of interest. And that will allow it, when this diazarine comes up to it, to become a carbene, which will allow you to do labeling. But the other critical feature is these diazarines, the things you'll use for this labeling, they don't interact with blue light. And because they don't interact with blue light, that's fine. It's only when they encounter the photocatalyst on your protein of interest. All right, why did we want to do this? Well, we wanted to do it because we've been talking to Merck about this molecule called Keytruda. If you haven't heard of Keytruda, it's a great thing to go read up on. It's the #1 selling drug in the world right now. It's just about to be overtaken by Ozempic, but it's the #1 selling drug. It's been in 2 million patients, and it's basically being used to treat 40 different types of cancer. So why is it able to treat 40 different types of cancer? It's a really remarkable thing. It's basically, this is a very simple biological picture, is when you have a cancer cell in your body, it's really a foreign agent. But you have an immune system and immune cells that's job is to go and get those foreign agents, and they're supposed to kill the cancer cell. But the cancer cell is really sneaky. And what the cancer cell does is get this thing called PD-L1, and it's basically a receptor that basically signals to your immune system, I'm a normal cell, don't kill me. And that's called the PD-L1, PD-1 checkpoint shown right there. So what Merck did, they came up with an antibody that basically breaks that up so that your immune cell can recognize the cancer cell and kill it. So that's amazing, right? And that's why it works for so many different types of cancer. So what's the problem? Well, the problem is it only works for 20% of the population. And based on the type of cancer, that 20% completely changes. And so Merck didn't know why. So really interested in, can you change that number? So they asked us, could we possibly start to look at this interactome? Can we try and understand this to a higher level such that they can try and improve that number? So we set out to do this, and we basically took cells that had PD-L1, there's our antibody with our photocatalyst on it, and we performed the experiment. Now, I'm not going to go through and try and teach you what this box means with all these dots, but what I will tell you is when we got this data, the biologists at Merck who we're working with were incredibly excited for reasons I'll point out in a few moments. But everything we cared about is in the top right-hand corner of what's called this volcano plot. We know we were able to run controls, for example, where when we take cells that don't have PD-L1, you see nothing in that top right-hand corner. So we know it's not sort of promiscuous binding that's taking place. So why were the Merck scientists so excited? Well, they were excited because if you look at everything that was known at that time with PD-L1, from 25 years of immunoprecipitation, here are some interactors that had been inferred. From gene co-expression, over 85 publications, these are the things that were actually known. And using this micromap approach, we were able to replicate all of that, but more importantly, we were able to find a whole range of other interactors which we subsequently validated, and we could do it all in 1 week using one experiment, which was something we were really, really excited about. So this took off like gangbusters. A lot of other people started to think about this. But one of the things that many people asked us, have you really quantified really what this radius is around these molecules when you actually label them? So we went back and we looked at the original one, the one that Alice Ting had developed, this beautiful approach using Apex. And this is why you get so many false positives, because it's got a 220-meter radius of diffusion. There's another really nice proximity labeling one called Minisog. That comes in about 140 nanometers. And when we quantified the micromap, what was really cool, we thought we're hoping for 10 nanometers. And I hope you can see this. This actually came in at 4 nanometers. So this is why you're getting such a good level of precision and you're avoiding all those sort of false positives along the way. Okay, I'll finish off, just tell you some of the things that we've done this and some of the other things that you could do with this. This was a paper we published a couple of years ago. This was really nice. This was basically, it was been known for a long time that basically in histones, in your chromosomes, and your histone, which is basically a wrapped up chromosome, if you have a one amino acid mutation, just one amino acid is changed, you will develop very quickly aggressive stomach and esophageal cancer based upon a one amino acid mutation. And people knew that was the outcome, but people didn't know why. So using micromapping, we were able to go in and tease out all this biological mechanism. And basically what's happening is you're losing an interaction with a deacetylase, which then, because it's got all these acetyl groups, it recruits this thing called a bromodomain, and you have runaway cancer growth. That's exactly what's going on. The good news about that now allows you to target that in a therapeutic way, which is one of the things which is ongoing. The second one was thinking about getting things into the brain. It's very difficult often to get molecules into the brain, and obviously there's many diseases associated with the brain, neurological ones obviously. So getting things into the brain is a big, big objective for the next decade for most labs who are in the biological realm. So one of the ways you can think about this is try and understand, can you get peptides in the brain? Now, as it happens, the vast majority of peptides don't go in the brain, but there's the odd ones that do, and people don't know why. So we were able to use micromap to tease out, it turns out there's 3 key receptors involved in bringing these peptides into the brain. You can now use that information and invent new peptides, which are even better, and now you can use those to effectively act like a small tugboat, bringing a sort of massive ocean liner into the harbor in the same way by, for example, taking these little peptides and using them to drag, for example, Taxol or antibodies, or basically anything you want and pull it right into the brain, which is something, again, we're very excited about. The last thing, and this is one of the things that I really think is cool, is we're able to think about why some patients respond to therapies and other patients don't. Another basically very successful drug is Herceptin. It was developed by Genentech. It's used to treat HER2-positive breast cancer. It works for about 30% of HER2-positive patients, and it fails for 70%. 70% are naturally resistant. So based on this, what we did was we simply took Herceptin and we put the photocatalyst on it, and then we mapped it. And we mapped it to see the difference in the interactome between the ones that are working, or the people who are susceptible, and the ones that are resistant. And we found there was one key receptor which was causing it to be resistant. The good news about that is you can then take a small molecule to block that receptor, and all of a sudden, Herceptin starts working in those cells again, which is, again, another area that's been developed pretty quickly. All right, I will finish off there. I always finish off thanking my lab. This is my group over in Princeton. About 2/3 of them are doing the chemical side, and about 1/3 of them are doing this chemical biology side, but amazing group to work with. I'll thank all these funding agencies, both from profit and non-profit, a lot of funding from pharma companies, which has been critical, especially now, and the funding from these non-profits. And then lastly, I'll finish off by thanking everyone here. You know, to come in here as an interloper, it's not always, you're always wondering how it's going to go, but people have been remarkably friendly, they've been remarkably nice. But more than anything, it's just been super cool to find out what you're all doing. And it's sort of blown my mind in a lot of different ways. So I will finish off by saying I love chatting with you, and I hope to have a lot more discussions with you while I'm here. Thank you so much for your attention.
David, thank you for a fantastic talk. It has been really interesting, and I think also a very nice thing to see that, I mean, you didn't just talk about the Nobel Prize, but about, even more coming afterwards. So we have time for questions. So I see Dave, can you put then?
That was a great talk. A lot of us in the United States are worried about funding. Given our NIH has played this important role, is it affecting your lab already? Yeah. What do you want to say?
Yeah, so I'll be very upfront. I'm very honest about it. We went 7 months with no federal funding. And so that cost us about, I have a big group, so that cost us about $1 million that we had to try and find from elsewhere. It was really pretty tough because, you're starting to think about how do you downsize, how do you start to do other things. We were able to figure out that problem, I have to say, and that's, This is a crazy thing to say. I mean, I could get out of it because, quite honestly, because I had a Nobel Prize. I could call people up and they would help me. But that's not the case for 99% of people in America. They can't just call up and say, give us money, and it somehow works out for them magically. It's a really, really tough situation. If you see the things that are going on in Harvard right now, and Princeton's got it kind of bad, Harvard's got it, Northwestern's got it incredibly bad. And you've seen all these labs up there which are downsizing. And these are labs which are literally doing incredibly important things, especially in cancer, especially in Alzheimer's. It's incredibly frustrating, not just for the fabric of what is America, but for the world. I mean, it's crazy. So yeah, I try my best not to go down that path, but it's hard when people ask you the question, but it's really, really tough. I hope it's getting better, but every time you read the New York Times, it seems like it's not necessarily getting better. That's a hard question to follow, sorry.
I'll change it to what's undoubtedly a silly question. Why blue light?
Why blue light? Blue light's great. Blue light is great because it's nice visible light, but it's quite high energy compared to red light and green light, so you get a lot more bang for your buck on the whole thing. And I always love to do this because I stand up and show up. Sometimes I have a blue laser pointer and I show blue. You can shine blue light on your hand all day long. It's not going to hurt you like UV light would, but it will put a molecule into this incredible energetic regime, right? And to me, that's what's incredibly exciting. The other thing that's really exciting, which I didn't quite talk about it, but I'll do it for a second. What's amazing about light is you can switch it on, switch it off. So for example, in biology, you can take a snapshot at 30 seconds or a minute or 5 minutes and you see the biology changing because it only labels when the light is on. So you get complete temporal control. But the other thing we're now playing with is using light with different light signatures. So some catalysts will take blue, some will take green, some will take other ones. And now you can actually stroboscope them so that they're compartmentalized in time. So they're only switched, because they only last about 10 nanoseconds when they're excited. So you can now have them only switched on for 10 nanoseconds, then the other one switches on, but they're never on at the same time. And so you can do very unusual selective things by stroboscoping the whole thing, all these different lights. So it's really kind of fun to sort of play around with that whole concept. Sorry, that was not your question, and I used it for more than you wanted, but yeah.
It was great, thank you.
Yeah.
Is there any other question?
Okay, Rosa Badia from Barcelona Supercomputing Center. Excellent talk, thank you very much. And thank you, Jacques, for suggesting this talk. So my question, I saw your drawings from your students in a notebook, and I was thinking, well, are you still doing this by hand, or how much are you modeling all your molecules in computing? And also in this meeting, we have had a lot of AI. I don't know how much are you using AI in your research? Thank you.
No, I have to say that's the number one. I say there's lots of questions people ask me. That's the number one question everyone asks you. Do you use AI and how do you use AI? Number one, we capture everything electronically now. All our data is electronic, and that was 1999, I think that notebook was. So that was all done back then. So we do everything electronically now. On the AI front, we do utilize it to some extent. The way that most chemists are using it right now is kind of boring, in my opinion. It's kind of for optimization. And optimization is really important, right? If you've got a reaction, you're making a drug for the world, and a step is 20% yield, you need it to be like 95% yield. How are you going to do that? You could spend a year futzing around in a lab, or you could basically get enough data to then have AI predict it. And AI is really good at helping you pull together the parameters and then find the right pattern to optimize it. That's really good. The thing, though, the thing that I do, and I think the thing that most chemists in my world do, is trying to invent new chemical reactions. And thus far, it hasn't been able to do that. So it's not been able to invent the next thing because it's kind of tricky to invent the next thing. You can give it all the information of what's known in the physical world, but then to predict what it can do with it, so far hasn't been so successful. As a short-term thing, and this is something that came up in the AI for mathematics and the AI for physical science panels, which I really liked, was I'd never heard it phrased this way, but the conjecture. And the conjecture thing is where I've been trying to preach or suggest to people, this is where we should be going with AI and chemistry. We have ridiculous sources of abundant materials on Earth that cost nothing. And yet there's other things which we use daily which are really difficult to get hands on, high energy involved to make them and use them. Why are we not thinking about, well, if you took this molecule from here, which is really abundant, and this one, and could somehow join them together, this would actually solve the problem. And then you let chemists think about, how am I going to do that? And someone will solve it. Because if you give a chemist a problem and say, solve this problem, they're pretty good at it, but sometimes the hardest part is the conjecture. And so I do think for conjecture, the AI part, the hypothesis part, could be absolute game-changing for the chemistry world.
Yeah, hey, wait for the mic, and then...
Please make remarks. Could mathematical modeling of the chemical processes help? We did, for instance, modeling for the photosynthesis. We did modeling together with Edel on his catalysis. But has it been done in your case?
Yeah, the modeling, we've used modeling a lot, and people use modeling a lot. The problem with modeling, modeling's a good thing. I shouldn't say the problem with modeling. A problem with modeling is that the way that modeling works is that you search for what the human inputs to say, we think it's doing this, 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 and this. Does this make sense? And you run a model and it looks good. And you say, oh, this is what's happening because it all makes sense. But it's not taking into account the other 100 things that could be happening that might have a lower energetic pathway. So it'll give you things which are consistent with, but it won't tell you that things are normally correct. Now, as you get back to more fundamentals, like if I wanted to predict a bond strength on a molecule, it's amazing at doing that. That actually is really useful, it turns out, especially for metabolomics and drug discovery. But if you're trying to figure out, am I actually going through the pathway I think I'm going through, you can get things which are consistent, but they might not necessarily be correct, and that can take you off in the wrong direction.
It only could help to make a test hypothesis.
Yeah, no, it's definitely useful in cases where people have the wrong hypothesis. And you can say, this is clearly not happening based on this modeling. And that's very useful. But when it actually comes to, the thing you got to be careful of is if you say, we think it's definitely going through this way, and then everyone runs in that direction, and nothing else works. And then you're like, it's a lot. So it's one of those things, it's the wild goose chase thing. So people are really big on trying to make sure, people would much rather you put out a paper and say, this is a hypothesis, than here's a computational model that we ran, and we believe it's this, because that's dangerous. But if you put it out and say, this is at least it's consistent with it, that's okay. So it's all about how you sort of couch it, I would say.
Thank you.
I think we have to stop with questions now, so you can approach David in the coffee break and over also in the evening. Last question. Thanks again for fantastic.